The chloroplast, like the mitochondria, is relatively large. In fact, chloroplasts can be even larger than mitochondria at 2 to about 8 micrometers in length. Chloroplasts are typically easily visible inside of the cells from green plants because they are loaded with the pigment called chlorophyll. The chloroplast is, of course, the site of photosynthesis, which is the metabolic process that harnesses the energy from sunlight and uses it to build glucose. Like mitochondria, chloroplasts have an inner and outer membrane, and the two spaces that we saw, both inside the inner membrane and between the inner and outer membranes. We're going to have some different vocabulary, though, to describe these membranes and these spaces. The inner chloroplast membrane actually gets a name. We refer to it as the thylakoid membrane. And instead of being folded up into cristae, the thylakoid membrane is even more highly folded, forming stacks of what look like pancakes that we refer to as thylakoids. The stack itself also gets a name. Those are called grana. The singular for grana is granum. The grana are actually connected to each other so that these stacks, these thylakoid stacks, are all connected to each other within that inner membrane. There is a space inside each of the thylakoids. That space gets a very general name. We refer to it as the lumen of each thylakoid. And the space that is between the outer chloroplast membrane and the thylakoid membrane is referred to as the stroma. This is a drawing of a chloroplast that shows some of these structures. In yellow, we can see both an outer and an inner membrane. It's a little bit deceiving because you can't readily see that the inner membrane is connected to these green structures. But in the actual cell, in the actual chloroplast, those two things are connected to each other. It's the inner membrane, in other words, that folds into these structures that are referred to as grana. You can see that each one of these structures is actually a stack of individual folded sacs called thylakoids. And each grana, each stack of thylakoids, is connected to each other. Now, the other thing that you can see in this drawing is that there are ribosomes inside of a chloroplast, just like there are ribosomes in the matrix of the mitochondria. But here in the chloroplast, the ribosomes are located in a compartment referred to as stroma. The stroma is also the site of chloroplast DNA and some starch granules. Remember, starch is a polysaccharide. It's a complex carbohydrate that is made out of strands of individual glucose molecules all connected to each other. Inside the chloroplast, as glucose is manufactured in photosynthesis, the individual glucose molecules can begin to be bound together to build these starch granules. So the chloroplast has the important job of being the site of the process called photosynthesis. And it's inside the thylakoid membrane that a pigment is embedded, the pigment known as chlorophyll. Light energy from the sun will be absorbed by the chlorophyll pigment. Remember that the chlorophyll molecules inside the chloroplast are arranged in groups that we call photosystems. There are actually two different photosystems inside of the chloroplast. There's photosystem 2, which is responsible for the production of ATP, and photosystem 1, that's responsible for the production of a molecule called NADPH. The two photosystems are connected to each other. The products of photosystem 2 and photosystem 1 are going to be used in a process referred to as the Calvin cycle. Carbon dioxide, which enters into the leaf of the photosynthetic plant, is going to be fixed and reduced to build the glucose molecule using the ATP and the NADPH that is produced during the first part of photosynthesis, called the light reactions. Now, it's important to know that in certain plants, there are other reactions that are going on in the chloroplast. For example, in some plants, nitrogen gets fixed and reduced in the chloroplast. You can see that reaction down here on the lower right-hand side of the slide. Nitrate will actually be converted into ammonium. Ammonium is a more useful form of nitrogen for the plant to use. Other plants are capable of fixing and reducing sulfate. You can see the reaction here, where sulfate is being used to produce hydrogen sulfide gas. Again, a more useful form of sulfate for the plant to use. So when we look at plants, particularly the leaves of green plants, under a light microscope, we're often taken aback by the number of chloroplasts that will be visible in the cell. On this slide, you're looking at an image of just an ordinary leaf from a green plant. And each one of these compartments here are cells. Remember, the cells from plants are going to look much more ordered and squared off than the cells from animals. And that's because plant cells have a cell wall around them made out of cellulose. So this right here, which I am outlining with the cursor, this is an individual plant cell. And I think you'll agree that this particular cell is loaded with chloroplasts. Each one of these circles is a chloroplast. You can't see any other organelle in here. You don't see a nucleus, you don't see any mitochondria, you don't see anything except these green colored chloroplasts. The reason that is because you don't have to do anything to this cell to see those chloroplasts. This is an unstained preparation of plant cells. We can see the green chlorophyll molecule with our eyes. We don't have to specially prepare the cell and stain the cell to see this pigment. If we wanted to see the nucleus, for example, we could prepare this cell and stain it specifically to see the nucleus. But if we're looking at it unprepared under the microscope, the only thing you're going to see are those very abundant chloroplasts. Now, there's one other structure in plant cells that I just want to mention briefly here, and that's the vacuole. Vacuoles are organelles that are found only in plant cells, and they have the job of storing water inside the plant cell. So in this image, you're looking at a vacuole taking up most of the space inside of this plant cell. The nucleus of the cell is located right here, and just about everything else that's visible in this cell is a chloroplast. Now this image is not colorized, so the chloroplasts don't have that nice bright green color that we saw a moment ago. But I think you'll agree that the vacuole is taking up most of the space in this cell, and that's because it is loaded with water. So three-dimensional macroscopic plants have to maintain their structure using what's called turgor pressure. It's the pressure of water inside the vacuoles that are within each of the plant's cells that give those cells their rigid three-dimensional structure and allow the plant to stand upright. When vacuoles contain less water, as when plants start to dry out, the vacuole will shrink up in the cell and the plant overall will begin to wilt. So vacuoles are critically important for maintaining, again, this turgor pressure that allows plants to stay upright. The endoplasmic reticulum is often referred to by the abbreviation ER. The endoplasmic reticulum is a very abundant network of membrane that sits inside of eukaryotic cells. This membrane, like a lot of the membrane that we see inside of cells, is flattened so that you get as much surface area as you can get without taking up any more space than absolutely necessary. This network of membrane is actually continuous with the nuclear envelope, the outer membrane of the nuclear envelope. So that tells us something. It tells us that the ER is going to be functionally connected to the nucleus. Now, the flattened membranous sacs of the endoplasmic reticulum get a name. We refer to those as cisternae. And they are also interconnected to each other by tubular connections within this network. There are two types of ER inside of cells. There is rough ER and smooth ER. Now, those two forms of ER got their names because of how they look underneath the light microscope. The rough ER looks rough because the surface of the membrane is studded with ribosomes. It is absolutely covered in ribosomes. Now remember, ribosomes are their own separate structure within a cell. They just happen to sit on the surface of the rough ER. Remember too that ribosomes are not organelles, and that's because they do not have membrane around them. The rough ER is the structure inside of the cell where protein synthesis and protein processing or modification is going to occur. The smooth ER is very different. The smooth ER was named because it doesn't have any ribosomes on its surface, so it looks much smoother under the microscope. You'll also notice that the smooth ER looks more tubular and less flattened than the rough ER does. The smooth ER has nothing to do with protein synthesis, even though it is continuous with the rough ER. Those two types of ER are connected to each other. The purpose of the smooth ER is primarily the synthesis of lipid and steroid, but it has a couple of other important jobs to do as well. Certain drugs are taken apart or detoxified inside of the smooth ER, and calcium is stored in the smooth ER. The release of calcium is tightly regulated. On this slide, we're looking at a drawing of the ER and its relationship to the nucleus. I really like this drawing because it shows us the difference between the smooth and the rough in terms of whether or not they're flattened sacs or more tubular structures. So take a look at what you can see in purple first. This represents the nucleus, and the artist has drawn it as though we've cut a section right through the nucleus and the ER. You can see that the outer nuclear membrane is connected with these little channels to the beginning portions of the rough ER, which is drawn in this magenta color. I think you'll agree that this is a complex network of membrane, and the membrane is flattened into these compartments or sacs. The entire surface of the rough ER has these little bumps on it. Each bump is a ribosome. Now, the rough ER continues directly into the smooth ER, but the smooth ER looks very different. Instead of having flattened compartments within it, it has much more rounded tubular compartments associated with it. Nucleus, rough ER, smooth ER. On this slide, we're looking at a transmission electron microscope image of smooth and rough ER. If you start over on the right-hand side of this image, you can see these flattened sacs of membrane. Now again, the way that ER is going to look inside an actual cell is going to vary. It depends upon the section through the cell that was collected to take the image. But I think you can see that each one of these sort of pancaked, flattened sacs of membrane has little tiny dots sitting on its surface. And those little black dots are ribosomes. So we know that we're looking at rough ER here. Now, over on the other side of the slide, it looks very different. This over here is smooth ER. Much more reticular, much more of a network over here on the left. No ribosomes are visible. The space inside the membrane is more open, like a tube, rather than a flattened sac. So rough ER on the right, smooth ER on the left, and you can see, especially right here in the center, that those two areas of membrane are connected to each other. They're continuous with each other, even though what's happening within the two types of ER is very different. So in the rough endoplasmic reticulum, on the surface, ribosomes are doing the work of translation. They are taking RNA and using it to make protein. That protein is going to be pulled into the compartment of the rough endoplasmic reticulum. Again, those inner compartments are sometimes called lumens. The protein is going to be processed or modified inside of the RER, and it will travel through that network of membrane as this processing goes on. One of the most important things that happens to protein inside of the RER is that it gets folded into its final three-dimensional shape. At the end of this network of RER, mature folded proteins are going to be packaged into membrane-bound compartments called transport vesicles. And those vesicles will bud off of the ER and travel through the cytoplasm to another organelle called the Golgi apparatus. Now we're going to be talking in detail about protein synthesis and protein modification in a couple of weeks. But for now, I also want you to know that there are some ribosomes inside of a eukaryotic cell that are not sitting on the surface of the rough ER. There are some ribosomes that float free of the RER in the cytoplasm. On this slide, you can see another TEM or TEM image of rough endoplasmic reticulum. Again, how the rough ER is going to look in any cell is going to vary, but generally speaking, you're going to see these flattened sacs of membrane that are stacked on top of each other. And on the surface of each one of these sacs is going to be ribosomes dotting it, making it look very rough on its surface. Now you can't see the nucleus in this image, so you can't see the connection between the outer nuclear membrane and the rough ER. But you can see a couple of other structures sitting here and here. This is, or these are, double membraned organelles that happen to be located, again, right here and right here. The inner membrane has folds. Any idea what these might be? These are mitochondria. These are mitochondria that are sitting within the cell and happen to have been caught in this section of the cell that was prepared for viewing.